So our next speaker, Cameron Macias, she has working on a very important project and on the, sorry, I lost my place. She grew up in a small town in Port Angeles in Western Washington State. She completed her BS in environmental science from the Western Washington University. Prior to attending the University of Idaho, Cameron spent 4 years working as a wildlife technician for the Lower Elwha Tribe, of which she's a member. She started at the University of Idaho as a master's student in 2018 and transitioned into the PhD program in 2020. Cameron enjoys gardening, painting, hunting, and has recently painting and hiking, goodness gracious, has recently become a super fan of the Vancouver Canucks hockey team. And she is going to be telling us all about the Cougars in the Temperate Rainforest, the Olympic Cougar project. So please take it away, Cameron.
Awesome. Thank you so much, Rachel. Okay, and may I share my screen? Okay, how does that look? Can everyone see my slides?
Looks great.
Awesome. So thank you for the introduction. Thanks for having me here. My name is Cameron Macias. I'm a PhD student at the University of Idaho, and today I'm going to be talking about counting cougars in the temperate rainforest as part of the Olympic Cougar project. So before we dive in, I want to go over an outline of what I'm going to be talking about today. So I'm going to start by introducing the Olympic Cougar project, why we started this project, why it's important to the Klallam Tribe to study wildlife. And because that's a bit of a mouthful, I'm going to just refer to the tribe as either the tribe or the Elwha tribe throughout the rest of this talk. And then I am going to talk about how we're estimating cougar abundance. And cougar abundance, what I mean by that is basically how many cougars are in the particular study area we're looking at, so population size. And that is part of the project that I am focused on as a grad student. And then after that, I'm going to wrap things up by sharing some of the other work that we're doing in addition to counting cougars. So let's jump right in. So the Olympic Cougar project is taking place on the Olympic Peninsula in the northwest part of Washington State. We're a peninsula surrounded by water on all three sides. So we're west of Seattle, south of Vancouver Island, and I am currently over here in the east in Moscow, Idaho, at the university. Okay, I think my slides are lagging a little bit, so sorry about that. Okay, so the Olympic Peninsula is the homeland of several tribes, including the Elwha tribe, which is the tribe that I'm a member of, and also the tribe that initiated this project. And it's really important for these tribes to understand wildlife trends in their historic use areas, because in 1855, these tribes signed on to treaties and began their federally recognized relationships with the US government. And the Lower Elwha tribe signed on to the Treaty of Point No Point in 1855. And so within this treaty, we reserved the rights to hunt on open and unclaimed lands, regulate hunting activity of tribal members, and to manage wildlife resources. And so we have co-management responsibility for wildlife in our treaty protected areas, and we set harvest regulations that differ from those set by the state, so by WDFW, Washington Department of Fish and Wildlife. And so it's important for us to know what's going on with our wildlife population. And so the overall goal of the tribe's wildlife program is to monitor and manage wildlife, as well as preserve intact wildlife habitat for future generations to enjoy as well. And so historically, we've studied deer and elk populations, as well as the other tribes on the peninsula. They've also studied deer and elk populations, but until now, we hadn't studied our predator populations. So we're missing really important information or data for setting tribal harvest regulations. And so in 2018, we started the Olympic Cougar project. And in total, this project has multiple goals. Today, I'm going to focus on the first one, which is estimating cougar density on the North Olympic Peninsula, specifically in the Lower Elwha Tribe historic use area. The second is to evaluate cougar movement and dispersal patterns. And ultimately, we plan to establish a long-term wildlife management plan for the tribe using data that we collected from this project. And so the project started as a collaboration between the University of Idaho and the Lower Elwha Tribe, and it was funded by a grant from the Administration for Native Americans. But shortly after we started the project, we teamed up with Dr. Mark Elbrock with Panthera and formed a formal relationship with the organization, which is a wildcat research and conservation organization. And since that time, our project has just expanded so much. And now we're actually working with multiple tribes on the peninsula. And the project is now co-led by Panthera and the Elwha Tribe. So our primary study area is the Pishtke Management Unit, which is just west of Port Angeles, and it's the core of the tribe's historic use area for hunting and gathering. And just to be clear, this is the Lower Elwha Tribe's historic use area. The other tribes that I mentioned a moment ago are doing their research on other areas of the peninsula. So if you haven't visited the Olympic Peninsula before, this is what it looks like. It's a coastal temperate rainforest with 200-foot trees. It's really wet and green, and it's super dense. And so it can be really challenging to count cougars. For example, here's a series of photos that each have a cougar in them in the tree. And even standing directly beneath them, I could still barely see them. And so I'll circle them so everyone can see where they're at. But they're incredibly challenging to count, especially in the Pacific Northwest. This one's really hard to see, but I'll point it out if it's large enough on your computer. This is a little face right here. That's the hardest one to see. So how are we counting them? Well, before we dive into our counting methods, I just want to take a moment to tell you a little bit about the cougar. They're large carnivores. They're the fourth largest cat species in the world, and they're the largest cat that can purr. They're mostly solitary, but they interact with their neighbors with frequency and some predictability. This is a just a little interaction. They form really intimate family bonds and stay with their mothers for up to 2 years before they disperse to find their own territory. They're really playful. They're just like big house cats. This is a kitten in our study area, and it's not cute at all, right? So cougars have the largest range of any terrestrial mammal in the Americas. And because of that, they are the world record holder for most common names because they stretch across such a vast area and across so many different cultures. So when you hear cougar, mountain lion, Florida panther, puma, or catamount, for example, these are all the same animal. And my tribe calls cougars sap. So cougars play a really vital role in supporting the health and function of ecosystems. And in doing this, they promote biodiversity. So when cougars kill their prey, I'm not sure if you can see my cursor, but if you can, when cougars kill their prey, the nutrients left over from the kills spread out throughout the ecosystem and help strengthen the entire community and improve the overall health and resiliency of the ecosystem. So when cougars kill their prey, it basically helps fuel the entire ecosystem and food web. So counting cougars is really challenging. So we are using a combination of 3 different methods. We're using GPS telemetry from radio collars, genetic sampling, and cameras. And so the primary goal is to estimate cougar abundance or population size in our study area so that we can have a baseline estimate, because we've never estimated this number before. And a secondary goal is to identify which method we should use for long-term monitoring. So we're going to compare these estimates from the 3 different methods, and get a sense for how feasible cameras are as a method by themselves to come up with population estimates, because cameras are a really cost-effective method that we could use for long-term monitoring for the tribe if the results are comparable to the standard methods that are currently used. So our first method is telemetry using the GPS collars. And so we've been working with local houndsmen to treat cougars since 2018. So once they're on a tree, we use a drug called BAM to anesthetize them. We estimate their age, we take biological measurements, and we equip each cougar with a collar that collects GPS locations every hour. And then using the GPS location, we're going to use auto-correlated kernel density estimate to estimate the home range sizes of every individual. And then using these home ranges, we're going to overlap them to estimate, to get a minimum count of how many individuals are in this area. So this is our first method. The second method we're using is non-invasive genetic sampling using SCAT. And so non-invasive sampling means that we don't have to capture the animals or interact with them directly. We can collect data from them non-invasively. And SCAT is just another word for a fecal sample. So we worked with scat detection dogs to locate and collect cougar scat in our study area each spring from 2018 to 2020. And so in our first year, we worked with conservation canines. And in our second and third year, a group of handlers and dogs from conservation canines left and created their own group called rogue detection teams. It was the same awesome dogs and handlers. And so they surveyed our city area in the last 2 years, and they were really great. So for the scat surveys, we divided our study area into 32 16 square kilometer cells. And each scat team, which is 1 dog and 1 handler, would go out and cover 1 cell per day. And every time a dog would locate a scat, it would sit down next to the scat and wait for it to be collected by the handler. And then it would be rewarded with tennis ball play. So these dogs are really reward driven, and they seem to love their jobs because every time they find the correct type of scat, they get to play. So it's really fun. But how do scats help us count cougars? So once we collected the scats, I took them back to the University of Idaho to conduct genetic analysis. So when an animal defecates, it sloughs off cells onto the outside of the scat. I'm able to collect those cells by taking little subsamples or little pieces from the outside of the scat and transferring it into this preservative solution. And then I can extract the DNA. And this is a simple diagram of a cell here on the top right. Although this is actually a plant cell. Just imagine that the chloroplast is not there, and now it could be a cougar cell. So during the DNA extraction process, we break open these cells to access the DNA. And then through a series of steps, we're able to wash away all of the other cell debris and have the DNA bind to a filter, so that after we wash everything else through, the only thing left is the DNA collected from that scat sample. So it's a pretty cool process that was simplified, but you can see the little diagram here, the different steps. And next, using that DNA that we just extracted, I can identify the species that left the scat. So in this case, confirm that it was a cougar. And then identify unique individuals. And it's like taking a fingerprint of a cougar using its DNA. So once all of the individuals are identified, we are going to use a statistical method called genetic spatial capture-recapture to estimate cougar density. And our third method is cameras. So we randomly deployed 74 motion-triggered cameras across the study area each summer, starting in 2019. And we have collected hundreds of thousands of photos during that time. And so here we have a selection of photos taken from the cameras covering a full suite of different critters. but going through each of the thousands of photos one by one is really time consuming. And so we're working with an image classifier program called Temperate IDS, and it uses artificial intelligence or machine learning to classify images. So over the past couple of years, we've used thousands of images from the Pacific Northwest, or from the Olympic Peninsula, to train the classifier on these specific animals. And it's teaching the program, okay, that's an elk, that's a bear, that's a coyote, et cetera. So ultimately, when we get thousands of photos each year, we don't need to manually look through all of them. We can put them in the system and classify them. So to estimate cougar density from these photos, we're using a statistical model developed by Moeller et al. Called space-to-event modeling, which uses camera data. So this is a more novel method that measures the space in front of each camera, or the camera's viewshed, and then looks at a snapshot in time across all of the cameras to estimate how much space it takes to detect an animal. So what this model will allow us to do is estimate how many cougars are in our study area based on snapshots in time. So we're not seeing an animal walk in or out of the cameras, out of the photo, or out of the camera's viewshed. And so this will allow us to estimate the population size, and by continuing to use cameras over time, monitor these populations using a non-invasive and cost-effective method. So this is really important for tribes because, like I mentioned before, we manage our own hunting regulations, and we want to make sure that we're setting sustainable harvest regulations and knowing what's appropriate for what's on the ground, what's going on the ground. So now I want to go through some of my preliminary results. And so for telemetry, we have collared 14 cougars so far in the Fish Game Management Unit from 2018 to 2021. This is an ongoing study, so we are going to continue collaring cougars through at least 2023. And the collective Olympic Cougar Project, so across all of the tribes, has collared over 60 cougars across the Olympic Peninsula since 2018. So we are really covering some ground out there. And for genetic species ID, here are the number of scats collected each year on the left, so between 150 to 300 about. And this is a map from the 2018 scat survey showing all the scats we collected. But this is our first year, so if we zoom in on the legend, you can see that we collected a lot more bobcat scats than cougars, 166 to 27 cougars. So, we started with this project with a focus on cougars, but we also wanted the scat teams to collect bobcat scats just in case that they were cougar, because scats can be really difficult to identify in the field. So very early on, we decided to also include bobcats in this study. We wanted to estimate their population size because we were collecting so much bobcat data, so we wanted to include that. So we identified 19 cougar scats in 2018, 38 in 2019, and 111 in our last year. So it's quite a big jump in 2020. And across all 3 years, we had a high amplification success rate for genetic species ID, which is great. It was about 90% each year. And for individual ID, we identified a minimum count of 6 cougars in 2018, 10 the second year, and 22 in our last year. And I just want to point out here that these are minimum counts at this point. They're not estimates yet, because we still need to run the statistical analysis before we will have actual abundance estimates. And we had a success rate of about 50% each year, which is typical for fecal samples when using microsatellite. And out of the 14 cougars collared in our study area, we detected 10 of them, 10 of the collared individuals with our scat surveys. So for cameras, out of the thousands of images, we had a total of 15 cougar photos in 2019. So we are still processing our 2020 and 2021 data, but the Panthera IDS image classifier is just about ready. So we're hoping to get rolling on classifying those images soon. But we do have phase 2 event estimates for 2019. The model produced a density estimate of 1.65 cougars per 100 square kilometers and an abundance estimate of about 10 for our whole study area, which is 606 square kilometers. And this density estimate falls within the average density estimated in the state of Washington in 5 different regions. So we are happy to see that. And so large carnivores are really hard to count. And it's rare that we can use multiple methods in the same ecosystem for the same study. And so we're in the early stages right now of planning to use all of those methods to build an integrated model where rather than just comparing them, we can also use data from multiple strains to build stronger estimates. So these will be integrated spatially explicit capture-recapture models. And ultimately, we're trying to sort out which one of these methods or multiple together will be the best to move forward for the tribe. These are, all 3 expensive and time-consuming methods. Managers are always looking for ways to improve on these methods. So we're really hoping that this camera-based model is going to be a big improvement if we can demonstrate that it is relatively precise compared to the standard methods that are currently used. And this project is really important for the Elwha tribe. It'll provide the first cougar estimates and bobcats in our historic use area, as well as provide the first estimates for the other tribes on the peninsula who are now collecting data. And this will help us to identify long-term monitoring methods for the tribe, like I mentioned. So once we get our results and finish analyzing the data, we will be able to move forward with a long-term plan. So now I just want to quickly talk about some of the other goals of the Olympic Cougar project. Using the GPS collars, we're tracking cougar movement and dispersal on the Olympic Peninsula. We know that over time, the Olympic Peninsula will grow as people continue to move there. It'll likely become a climate refuge as other areas of the country become warmer. So we want to understand the resources that are important for wildlife. So with cougars, we're looking at their home ranges and especially subadult dispersal patterns. And the reason this matters is because, like I mentioned before, the Olympic Peninsula is essentially an island surrounded by water on 3 sides. And then we have the city of Olympia, we have the I-5 corridor to the east, and then the Columbia River to the south. So it's pretty hard for wildlife to get on and off of the peninsula. They either have to cross I-5 or swim across the Columbia, which is not an easy thing to do for animals. And so we really want to know, especially for wide-ranging species like the cougar, whether we're seeing any connectivity between the Cascades and the Olympic Peninsula. And we know that we are among the lowest genetic diversity in North America, along with the Florida panther. The cougars on the Olympic Peninsula have low genetic diversity, presumably because they're not able to get off the peninsula very often. So we are focusing on learning more about that connectivity and dispersal. So this is a home range map that shows 21 of our collared cougars from across the peninsula. So like I mentioned before, the collective Olympic Cougar Project has collared over 60 cougars since 2018. And here we're showing home ranges for 21 of them using 65,000 GPS locations. So we have both a minimum convex polygon in the lighter blue and a kernel density estimator in the dark blues and pinks. So we're really showing here that we're starting to cover the peninsula in cougar movement data. And we're also able to look at dispersal patterns. So looking at the young females and males at about 18 months old that leave their mothers, we're seeing some cats disperse pretty far down south and up north on the peninsula. We have one cat that actually did cross I-5 for about 2 days and then came back. And then he went down to the Columbia River and then turned around. So this is incredibly valuable data. And we're going to continue looking at dispersal as we continue to collar more animals. And so another thing that we can do with the GPS data is we can figure out what these animals are doing when they stay in one location for an extended period of time. So the box in red is circling what we call a cluster of locations. And so we hike into these clusters to see what the animal was doing there. Was it a bed? Was it a den, a mating site, or a kill site? And when we hike in, if this cluster is a kill site, like the one here, this is an elk kill. If there's more meat left on the animal, then we, can assume that the animal or some other animal, maybe a scavenger, will return. So we put up a camera to see what comes back, and we record some pretty neat information. And lastly, I just want to touch on bobcats really briefly. We are also just starting to color bobcats. So my field crew, the ones who were in Port Angeles, while I'm over here in Idaho, have colored 5 bobcats now since the beginning of the year. And so now we have these animals on the air collecting GPS data points, and we're going to use this telemetry data to build an integrated model for bobcats as well, and hopefully get more precise density estimates with the integrated model. And so we're really looking forward to collecting more data on these wildcats and being able to share more results soon. And with that, I'd like to acknowledge everyone who's helped on the project, the Lower Elwha Wildlife Crew and Panthera, who are out in the field catching cats and deploying cameras and hiking into these clusters, the Wait Lab at the University of Idaho for helping with all the genetic work, and a huge thanks to all of our funders. And thank you all so much for attending my presentation. Here's my email address if anybody wants to reach out with questions.
Thank you so much. That was fascinating. I really appreciate learning all of that. There are people with questions, but I'm just going to ask about 2. One is the project a part of a 501c3? And so how do people support your work if they would like to financially or advocacy wise? Maybe we can put that in the chat for folks to look at.
Sure. Okay. Do you want me to respond in the chat?
Well, you can respond now and then we'll put it in the chat. How about that?
Okay. Well, what I would do is I would reach out to the project manager, which is Kim Fager-Fadkin, and I'll put her email address in the chat, and I'll also put Dr. Mark Elbrock's contact information in the chat as well. They are the ones who work on all of the funding and budget stuff, so I would connect you with them.
Thank you. Okay, I'm just going to ask 2 more questions. Who has access to the GPS data that you collect?
Right now, it is not public information. It is just, it's private information right now while we finish analyzing the data. And I'm not sure that that'll actually become public information just for the safety of the animal.
Thank you. And then the final question is, do you have any theories as to why there's a big jump in the cougar number estimates during that year, 2 years?
Mm-hmm. Yeah, so in our 3rd year, we collected our scat samples for 2018, 2019, and then in 2020, we saw that big jump. And we have a couple ideas about why that might have happened. We don't necessarily think that it has to do with an increase in the cougar population necessarily. Right now, what we think was going on is the way that the scats were being collected by the scat detection team changed a little bit. We had 2 senior handlers out, and they asked questions about where we were seeing cougar activity because they thought that the way that the cougars were using the landscape might be different than other areas where they were used to doing their surveys. And so areas where they usually would go look for scat, because the dogs kind of lead the way, right? They're smelling scat in the air and they kind of lead the handlers. They set out on that path, and the areas where the handlers were sort of guiding the dogs, lions, areas where they expected to see cougars, they just weren't finding much scat in the first couple of years. So they found a lot more scat samples in the final year, which gave us a larger sample size.
Thank you. Great. Well, I don't want to, I know we have another speaker coming up and we're very excited about her as well, but that was really wonderful. Thank you so much for your time and your work on this project. It was really interesting to learn all about that.
Thank you so much.